Hello, welcome back for another talk about microbiology. Last week, we talked about how bacterial cells are able to transform matter and energy. And this week, we're going to talk about what exactly they do with that matter and energy once they transform it and why they do it. And bacteria are the same as all other groups of living things. Essentially, they transform matter and energy because they need both in order to create new cells and more organisms. So just as a quick reminder, I want you to remember that I use the word biomolecule in place of organic compound because I like it better. It says it has bio in it instead of chemistry, but realize what they are and also realize that they are the primary sources that cells of all types use for both carbon and energy. So this is, I'm saying that because we're going to talk a little bit more about the transformation of matter and energy, starting with this. So here are lots of words. These are categories that are based on patterns of growth. I'm sure from your previous eukaryotic biased courses in biology, you remember the terms autotroph and heterotroph as producers and consumers. And what that actually refers to is autotrophs, which means self-feeding, produce their own biomolecules through a variety of different means, but they are the ones what they are doing is taking carbon dioxide, which is inorganic carbon, adding hydrogen to it, which makes it organic carbon, and we call that carbon fixation or fixing carbon. So autotrophs are the ones who are able to make their own biomolecules, and heterotrophs consume those that are already existing. And then, of course, as Morpheus will tell you, there's this category in between, which are bacteria and archaea, which are considered mixotrophs because essentially they can do both. They are able to be autotrophs when they need to be. They are also able to be heterotrophic. If there is no way to make your own biomolecules, you can go ahead and eat whatever is ready and available at that time. So we talked briefly about Rhodopseudomonas, which is an example of that type of metabolism, of growth pattern, which is one of those microbes that basically is chemically able to do almost everything. So on thinking about these two groups, autotrophs produce their own biomolecules. And so in the days of your previous biology, you heard about the plants doing this. They used photosynthesis, grabbing sunlight energy and using that energy to fix carbon, add hydrogens to CO2. So those are photoautotrophs. Like plants, like algae, like many bacteria also have the right equipment. In other words, they have chlorophyll molecules, which allow them to capture energy from light. And then they use that energy as a way to take those building blocks of hydrogens and carbon dioxides, put them together to form their own biomolecules. So those are the photoautotrophs. But this is microbiology. So let's talk about this group also, the chemoautotrophs. So photoautotrophs use light energy and convert it so that they can fix carbon dioxide. Chemoautotrophs use chemical energy. So they are oxidizing or getting electrons from inorganic compounds from the environment. And they use electron transport system to produce ATP, and then that ATP is used to fix carbon into sugars and other biomolecules. That's a process known as chemosynthesis, to compare and contrast with photosynthesis. So there are these two groups of autotrophic organisms. Photoautotrophs you've heard lots about, chemoautotrophs because they're bacteria you probably heard very little about. On the heterotrophic side, those are the consumers, the ones that are using already existing biomolecules for their cellular needs. And there are two groups of those also. We talked about the lithotrophs last time. Lithotrophy meaning using inorganic, literally rock eaters. They are using inorganic sources as their energy. So they're taking those electrons, putting them through the electron transport system, producing ATP, meets their cellular energy needs doing that, but then realize that they still need an organic biomolecule to serve as a source of carbon, because cells are cells and all cells are built out of the same stuff, right? And then on the other side of that, we have chemoorganotrophs, which would be us and other heterotrophic organisms that consume whatever is available that has biomolecules, so that we could use that as our source of both carbon and energy. So that's us, that's many bacteria, that's all the fungi, that's you know, all of those other microbes as well. So this gives us these 4 groups, and I just, you know, want you to realize that the ones that are bacteria only, bacteria and archaea, can be chemoautotrophs or they could be chemolithotrophs, but there are no other groups of living things that have these metabolic capabilities that allow them to do those things. So that makes them interesting and more special. So then we get to the point of, what do cells do with all of their energy and biomolecules that they're either making themselves or they're consuming from other sources? And the answer is bacterial cells are doing the same thing that other cells do, which is they take all that stuff and they use it to build new cell parts, build new cells, and then go through cellular reproduction. So bacterial cells, like other types of cells, reproduce in a fashion where they duplicate their DNA, right? So DNA replication occurs first. And then the copies, the 2 copies of their genomes starts to separate. And as the 2 copies separate, a septum forms, and eventually that septum results in 2 completely separate, which are called daughter cells, 2 completely separate daughter cells. But this is a process where one cell divides into 2 cells. The cells are pretty much identical to each other because they have the same DNA, and they are pretty much identical to the cell that they came from. So the daughters in this case are genetically identical to one single parent. There is no mingling of genetic information, although that's a lie, and I'll tell you more about that later. But the point being, it's an asexual reproductive process. And what you see here is something called binary fission, literally to split in 2, which is what these what bacterial cells do, many of them. And this process is very well recognized and established because all of our fast-growing laboratory types of bacteria, the ones that we've been able to grow in culture, the ones we know pretty much about, mostly reproduce using this method. But as we have learned more about bacteria and more about the reproductive means, there's a lot more to this than what we thought even just 10 years ago. So this process, which is symmetrical, meaning you get 2 identical copies of the cell. There's also asymmetrical forms of bacterial cell division. For example, bacteria can bud. They can do this process called budding, which is similar to what yeasts do. They can fragment, which is similar to what algae do, right? So they grow these long filaments that the ends sort of break off. They form baocytes, which I just like saying that word because it means it's like baby cells. So they literally have babies, all of which involve multiple rounds of DNA replication, usually before the reproductive process actually begins. So you end up with more than just a few copies. You end up with several copies because the DNA gets copied several times before the rest of this process occurs. And then I also want to give a shout out to endospore formation here, because technically endospore formation is a form of cellular reproduction. The bacteria copy their genomes and then they just kind of tuck it away into a part of a cell. So we're gonna focus on endospore formation for just a second here, because you notice here's the process of binary fission using those FTS-Z proteins that was characterized a while ago. So we actually know how the bacteria produce the septum that leads to the release of 2 daughter cells. So endospore producing bacteria, such as bacillus, such as Clostridium, and a few other types, go through binary fission when they are active, healthy, lots of food in the environment. You know, they're reproducing, growing to larger numbers. But they can also go through this process known as sporulation, which is DNA replication. And then the DNA, so here's DNA replication. And then what happens beyond that is the genome, the copied genome, gets placed into the cell. And once that happens, the FTSZ proteins start to form, and they form around the genome of the bacteria, and they encase it. They start the encasement process into a fully formed spore, which of course you remember from our staining experiments that you can actually see those inside of the bacterial cells. So this is asymmetrical type of cell division because you don't end up with two identical daughter cells. You end up with a dead, disintegrating body and an endospore. And then the endospore can sit there dormant for millions and millions and millions of years, and then germinate into a single bacterial cell, which can go back into the binary fission process. So technically, it is cell division. It is cellular reproduction. It's just in a way that nothing else has ever seen before. So that's sporulation. And then I wanted to point out that there's a bacteria named Apulopicium, which was discovered in 1985, by the way, which was a huge discovery at the time. I can reveal that I was around in microbiology at that time, and we were all like, oh my God, it's a giant bacterium. That's not supposed to happen. In fact, it was mistakenly thought to be some kind of protozoa. That's how big it is. So it's a giant bacteria, and they thought it was, you know, some kind of protozoa until they realized it didn't have a nucleus. And then when they looked at its DNA, they realized it's a prokaryote. And then when they looked closer, they realized it's closely related to these endospore forming types of gram-positive bacilli. So this is an endosymbiont that lives inside a fish. And you can see that this sporulation process is repeated with the reproduction of this Apulopicium. So it's endospore fashion, but notice here's the offspring that essentially grow into new cells and then are born, if you will, out to keep the cycle going. So again, another sort of unique approach to bacterial cell reproduction. But this is asymmetrical cell division as opposed to binary fission. And we're going to focus on binary fission because that's what we know most about. So we're going to go from here to talk about the process known as binary fission. Now, down below, you're going to see, while I'm talking, you're going to see bacterial cells going through time-lapse photography, showing a population that started with 2 cells. And notice that after 8 hours, you now have thousands and thousands of cells. And that is because when cells are reproducing by binary fission, the number of cells will double once every generation. And many bacteria, that time it takes, that generation time or doubling time, is a very short window of time, as you can see here. So in the 8-hour time-lapse frame that you're watching, we went from 2 cells to thousands of cells. And that's because at every division or every generation, the cell population is going to double. So if you start with 1 cell in the first generation, you will actually, the original generation, the parental generation, the first generation, there'll be 2 identical cells. And then each of those will divide in 2. So now we have 4. And the next generation, each of those 4 cells divide into 2. And now we have 8. And then we have 16. And then we have 32. And then we have 64. And then we have 128. And then we have 256, and I'm going to stop because now I'm going to have to think too hard to come up with the rest of the numbers here. But that's why after 8 hours, we go from 2 cells to thousands of cells because bacteria reproduce using this process known as binary fission. So what I want to point out is that not all bacteria reproduce at the same rate. So Escherichia coli holds the record, if you will, for the shortest doubling time, which is 17 minutes. So if you feed E. coli everything a growing bacteria could want, you give it everything it wants, give it dessert, give it a great environment, one bacteria, one E. coli cell can reproduce into 2 cells in 17 minutes. So think about that. Every 17 minutes, the population of E. coli is going to double. So that's like the world record as we speak right now. But notice as we go down this list of other types of bacteria, very common. Bacillus is a gram-positive bacillus. These are streptococcus, staphylococcus. Notice the times are not the same. So even given their happiest medium, there's quite a bit of variability in how long that doubling time is. And that's part of the strategy, their lifestyle and strategy for survival. E. coli strategy is to grow as quick as you can and spread as fast as you can. Same with many other types of bacteria. But as we work our way down, notice we get to Rhizobium, which is a plant, a bacteria that inhabits plants. We're talking, hours for one generation to occur. Mycobacterium tuberculosis, which is the causative agent of TB, also hours for those cells to reproduce, and they can go through very long periods where they don't reproduce at all. Even if everything is right, they won't reproduce. And then Treponema pallidum, which is the causative agent of syphilis, it's a spirochete, which generally speaking, spirochetes are not that easy to grow in a culture environment where we can control what they're eating and what they're doing. They tend to do what they want. And so you'll notice, though, on the other side, spirochetes tend to have a record for the longest doubling time. It is not their strategy to grow quickly and spread as fast as they can. They are more along the lines of, this ain't a bad place. I think I'm gonna, hang out here, build a little city, stay around. And if necessary, when the time comes, I'll make more of myself so that I'll be able to spread to new hosts. And the means of spread, the way they are able to spread, also, the slower growth rate is more conducive for those types of pathogens. So that's why, TB, people can be infected with TB for years before they actually ever experienced any kinds of symptoms of TB. Same with syphilis, same with Lyme disease, because that's the nature of those types of bacteria. So I just wanted to point out that this generation time or doubling time isn't the same across the board for all different types of bacteria. So what we do with this, looking at growth rate, we are able to create something called we can plot the growth rate, right? So we can look at the number of cells over a period of time. And because bacterial numbers grow so quickly, we're going back to that rapid reproduction and binary fission. Because they reproduce so quickly, notice that, after 1 hour, kind of as you look across the graph, you'll notice that in the beginning, the first several hours, it appears that the numbers of cells aren't increasing all that rapidly, but then suddenly you hit a point and the numbers start to increase very, very rapidly. And so they go, from 10,000 to 100,000 to 1 million. And that gives us this curve. And so what we do, because we want to infer information from this curve, is to, instead of plotting on the graph, to do the actual number of cells, which goes from 0 to 1 million in a few hours, we use a log phase. So log base 10. So we can plot the log of the number of cells over time, and then you get a nice linear graph. So this is easier to get data from. Like, for example, if I wanted to know how long it took for a population to double in size, all I need to do is find 2 points on this straight line that are double the number, and then go back and figure out how much time that took. So it's an experiment that you could do if you so chose to do. So most people use this approach, using the log base 10 over the number of hours, to produce what is referred to as a bacterial growth curve. So we're plotting the log of the number of cells over time, and you can see that growth of a population of bacteria has discrete stages that can be delineated from one another. And those stages, starting here, the lag phase, and realize that all of the time that each bacteria spends in these phases can be variable across the board in terms of species and, growth conditions. Is there enough food? So the lag phase, as you can see here, it looks like there's a lag in growth, meaning the numbers does not seem to be increasing or to any significant degree up till this point. And at this point, you can see that the number of cells goes into exponential growth. So in other words, this is the phase of growth where the number of cells in the population doubles every generation, right? So now that we're into that doubling phase. So during the lag phase, why does it look like nothing's happening? So, you do your subculture and you watch it for 8 hours. It's look like the broth is still clear. And then you go away and you come back in the next morning and the broth has become cloudy. That's because you left before we hit this critical moment where we've reached enough numbers of cells so that when the number doubles, right? So from 100, we go to 200, then 400, then 800, then 1,600 cells. So it's sort of like your retirement account, right? When you're 25 and you're looking at your retirement account and you're like, oh, great, I've got $25 in my account. And then as you get older and you hit critical mass, that number compounds on itself and you end up with a slightly larger amount, or at least that's how it's supposed to work. So the same is true of growth curves. So this is called the log phase. It's the exponential growth phase. And how long the bacteria are in this phase varies. But generally speaking, you'll notice it ends, right? So bacterial populations don't just keep growing in number unless there are unlimited resources and a way to, take away any of the end products that accumulate that might be toxic over time. But for the most part, if you just let that culture go, so imagine a broth, we drop some bacteria into the broth, we watched it, nothing happens, and all of a sudden it starts to get a little cloudy, then it gets really, really, really cloudy, and that will continue for, several hours, and then the growth appears to stabilize, right? So the number of cells stops doubling, and it stays roughly the same. And that's referred to as stationary phase. And stationary phase of bacterial growth is very interesting. It's at this point where the cells are running out of nutrients. It's the point where, like I said, toxic stuff is starting to build up because they're metabolizing and excreting at the same time that they're consuming. And so some of the cells will start to die. Some of the cells will continue to reproduce. But what I want to say here, and we'll talk more about this in the future, is that in stationary phase, a whole bunch of genes get triggered in bacterial cells, the ones we've studied, are turned on to rescue the cells. And it's kind of like this period of, oh crap, I'm running out of food. All this stuff is building up. What am I going to do? I better turn this gene on. I better turn that gene off. Or better yet, I'm just going to turn all my genes on. And hopefully one of them will be the thing that keeps me alive the longest so that reproduction can resume again when conditions get good. And during that stationary phase, a whole lot of genes get activated that we know about that lead to downstream effects that, again, something that we'll talk about later. But it's, this response is sort of, it has been called the save our cells or SOS response, where they turn on all these genes, hoping for the best that, something will do the trick and allow them to survive just that little bit longer. And, that stationary phase can go on for a really long time. When you consider the length of time that I keep bacteria on a slant, all those bacteria are in stationary phase. And they're sitting there on that slant for, 3 to 4 months in between the times that I subculture them. So interesting how bacteria are able to do that. But, if nothing improves and eventually the culture enters the decline phase where the number of living cells starts going down, and eventually if there's no intervention, the population can die out. But it's interesting because populations of bacteria, cultures of bacteria that you think are dead, that have been long dead, under the right circumstances can sort of reactivate to come back to life, which is a very interesting characteristic of bacteria, as you might expect. So this is the bacterial growth curve and what goes on in a population over a period of time. So of course, because there's graphing and numbers, there's math, a little bit of math. And the math that I want to talk about is just that we can express mathematically this idea that during binary fission, bacterial cell number is going to double every generation using this expression, right? So 2 to the n refers to the generations that have gone by. 2 to the n refers to the fact that every generation the population doubles. So how do we apply this? Let's look at this one, right? So E. coli, our good friend Escherichia coli, the winner of the fastest reproducer. So I'm going to give it 20 minutes because that makes it a little bit easier. So it's already been shown, we've already done these experiments, so we know that E. coli has a doubling time of about 20 minutes. So that means every 20 minutes, the population doubles. So if you started a culture of E. coli from one single cell, how many cells would there be if you let it incubate that culture for 6 hours? So the question is, there's what we know about the cells and the numbers. So after 6 hours, how many bacteria could you expect there to be in that population of cells if you start with one? So the answer is, how many generations have occurred? Because what we need to know is this little n. So 2 to the little n is how many generations have occurred. So think about this. The doubling time is 20 minutes. How many generations can occur in 1 hour? And the answer is 3 can occur, right? So 20 minutes, then 20 minutes, then 20 minutes. That's 3 generations of E. coli. And so if we multiply that by 6 hours, 18 generations of E. coli occur in 6 hours if the bacteria are growing in log phase, right? So that means now we know the number of generations, so we can say n, the number of cells in the population, is equal to 2 raised to the 18th power. And grab your calculators and do that, but I did it for you. That's 262,000 E. coli in 6 hours, starting from one bacterial cell. So here's another way of thinking about it. So at lunchtime, there was 1 E. coli in your undercooked burger that you just finished eating. At dinner time, 5 or 6 hours later, you're going to have 250,000 of E. coli in your GI tract. By the next morning, there will be billions of E. coli in your GI tract. And, I chose E. coli just because it was it's the fastest growing, but there are pathogenic strains of E. coli that can cause disease, and realize that E. coli is representative of the growth rate of other bacterial pathogens that cause GI tract illnesses. So we go from 1 cell to billions of cells in an overnight period when the generation time or doubling time is fast enough. So here's another really interesting thing about all of this. So 1 E. coli, you eat 1 E. coli. You're gigantic. Who cares? You have 1 E. coli, it's not going to kill you. Even if it was producing toxins, they're producing 12 molecules of it, it's not going to be toxic. But then you hit 250,000 E. coli, and if they're all producing toxins, now you have 250,000 times 12 toxin molecules, and suddenly that's a lot. And as the population increases, it turns out that they don't want you to know that they're there. So they will hold off on producing anything that might make you feel sick until there's enough of them there for it to make a difference. And that's something called quorum sensing. And so in the real world, it's important to realize that, the culture world, we can do all those fun experiments. We can create growth curves. We can say the log phase is going to last this amount of time. But in the real world, bacterial growth doesn't follow those culture-based rules. I mean, the lab is really very much an artificial environment when it comes to bacteria. So in the real world, bacteria grow in communities, and one type of community is called a biofilm, and it's like a huge metropolis of bacteria that are built by the bacteria, and within the biofilm, they are talking and communicating with each other about many, many, many different things. So here's how biofilms are known to form. So this could be E. coli, or it could be Salmonella, or it could be Pseudomonas, it could be bacteria that are known to produce biofilms. So first they find a place to settle down. They're the pioneers, right? And then they start to build their little houses, which are called microcolonies. Then they start to, the houses build up, and you've got like a general store shows up, and you've got more people coming, and the colonies start to get bigger, and then they take on a three-dimensional form, and they are this huge structure called a biofilm. So there are many places in your body where bacteria live in biofilms, and that's the way they survive. So your teeth, by the way, your teeth and gums are one place where you've got a bazillion bacteria growing in biofilms. Once they're in the biofilm, they are very difficult to remove because it's a city and it's protected, and they talk to each other to make sure that they protect themselves. And if they get the right signal from their environment, they will release new planktonic bacteria off to be new settlers of different communities to keep the population growing. So these are referred to as planktonic bacteria because they're the ones that are swimming around and moving around. Once they hit biofilm phase, they shut off all those other genes, the motility genes and everything else, and they devote themselves to this biofilm type of community. And I just want to point out, because I must, that in a biofilm, when you think about it, it's a multicellular community of bacteria where they are communicating with each other, each carrying out different tasks, if you will. Sometimes one of the tasks is to make the host sick, and by that I mean, think about going back to E. coli. So you eat E. coli, then you've got a billion E. coli, and then they all start to produce toxin genes, for example. Those toxins, when they hit critical mass, will make you very, very ill. So you will have diarrhea, for example. A great way, if you're a pathogen, to spread to, at least 35, 40, 50 people that are associated with the person that has the diarrhea. So it is their strategy. They grow to a point, they hit those toxin genes, they say to each other, all right, is everybody here? Turn on the toxin genes. They release a toxin that causes your physiology to dump water into your GI tract because the toxin says, get that out of there. That causes diarrhea, and now they're all over the place outside of you where they can be picked up by other hosts. Isn't that brilliant on the part of the bacteria? So multicellular behavior in a bacterial colony, bacteria are referred to, of course, as unicellular, everybody's, they're their own entities. They aren't. They live in communities, they do multicellular things, and there are those people who will argue that multicellular life originated from this type of thing. One of them are these people right here. This is Bonnie Brassler, who is at Princeton University, who studied what I just told you about, the idea of autoinducers. Autoinducers being the chemical signals that bacteria and biofilms use to say, okay, everybody here, all right, now everybody turn on the genes for the toxin. So it coordinates behaviors, often about virulence, what makes them dangerous. And the human, the health, the medical aspects of biofilms as a result of that is discussed in this particular video on YouTube also. So I'm not going to show you those, but please go watch both of these two. This one is 6 minutes. It will provide you with a medical perspective on health perspectives related to what biofilms, what we know about biofilms now. And Dr. Bressler is just going to give you, again, an 11-minute talk on her research showing that these autoinducers were a way that bacteria are able to speak to each other, and that biofilms are actually multicellular life, representing multicellular life on the prokaryotic level. Okay, so when you get a chance, go watch those. Okay, so that's how they grow. Now let's consider some of the things that are important to support growth. So what does a growing bacteria need in order to survive? In other words, what environmental factors are important here? And just like every other living thing, biomolecules are key, because why? We need a source of carbon and we need a source of energy, right? Matter and energy, transformation of matter and energy. Why do cells transform matter and energy? Because they need to make new biomolecules so that they can build new cells so that they can go through cellular reproduction. So that's kind of key, and that is actually what we talked about when we discussed metabolism, how they go through those processes. So you got to feed them, look at it that way. You got to feed them, and then you got to nurture them. So you want to make sure that the environment you're growing them in has the right stuff to support their growth. So there are many other chemical and also physical factors that we we concern ourselves with when we're trying to culture bacteria, right down to the incubators that we put them in when we want a culture to reproduce so that we can do something with it the next day. So one of those chemical factors is oxygen, which we've talked about before. Oxygen or alternative electron accepting gases like nitrate, like sulfate, like carbon dioxide for the anaerobes. So in other words, the atmospheric conditions, the gases that are present in the atmosphere are important. So if you have an aerobe or an organism that might be anaerobic but can live aerobically, you're fine growing them in a regular incubator. But if it's an anaerobic bacteria, you would need to have a special incubator where the oxygen is gone, but enriched with these other gases as well. So it's a consideration for growth. You also want to grow them at their permissive growth temperature. So not everybody is like us that prefers 98.6 degrees Fahrenheit or 37 degrees Celsius. So especially on the microbial side, there are microbes that like it colder, there are microbes that like it hotter, and there are bacteria that like it boiling. So you would have to take that into consideration. And then you also have to take into consideration the osmotic concentration, osmotic pressure. Water pressure is that's a pressure that all cells face all the time. And for example, our cells, we work really hard to keep that concentration of solute in water roughly about the same in our blood, so that cells don't end up under osmotic pressure and end up bursting or end up shriveling up or dying, because that's one of the things that happens here. And then the appropriate pH for survival. So, neutral pH 7, is what a lot of people think, oh, you have to have a neutral pH in order to survive. Well, we do, certainly, but microorganisms do not. And there are, they can live across all pH ranges, which is interesting too, even within our own bodies. So, our bodies are buffered to keep a pH of about 7.2 to 7.6, but there are places in our bodies which are not 7.4 all the time. Like, for example, in our stomach where the pH is 1. Well, surprise, you have bacteria growing in your stomach in that very acidic environment. Likewise, other parts of our body, the pH is slightly higher, like in our urinary tract. So we have bacteria growing there that prefer that slightly higher pH range. So where to begin? I'm going to start with oxygen, and just kind of because we've been doing this in lab quite a bit, talking about the idea that you have to provide a final electron acceptor for the electron transport system so that cells can make ATP. So realize that if you're going to live the aerobic lifestyle, there is a price to pay. And that would be the price being the formation of reactive oxygen species due to chemistry. So what does that mean? So here's my little, representation of an electron transport system in a membrane, right? So here's all those redox enzymes. This would be cytochrome oxidase, hopefully you remember that. And this would be ATP synthase, which is how that ATP gets made, right? This is the chemiosmosis part of it. We're going to leave that off. Electron transport, we get to cytochrome oxidase. What happens? The electrons flowing through the electron transport system end up with oxygen and water forms. Yay, so oxygen to water. Water is not reactive. Water is important because we actually need the water for all of this chemistry to occur. So we've already talked about water. But realize that chemistry doesn't always do this exactly right. So we can end up with what are referred to as reactive oxygen species. So what are these things? They're these guys. So this is a sneaky thief, and these are all sneaky thieves that are going around grabbing electrons from other places. So reactive oxygen species are going to result in the oxidation of things that we don't want oxidized, like lipids, like proteins. And you may recognize a couple of these, right? So in fact, peroxide, right? So we have enzymes called catalase that help break down this particular reactive oxygen species. Superoxide anion, we have something called superoxide dismutase, which is another enzyme that breaks down these reactive oxygen species and others. Because if we're going to live the aerobic life, we have to deal with this stuff. And so we are able to deal with this stuff using all these different enzymes. So not only do you need all of the electron transport enzymes and all the rest of that stuff, you also need enzymes that allow you to take care of all of this, this toxic stuff, which guess what? Aerobic organisms do. So that would be part of their traits. Their traits are, we have to process this, so we're going to produce catalase, we're going to produce superoxide dismutase. So if you live the anaerobic life, it's a lower price because you'll notice you don't have any sneaky thieves being produced. There are other, I shouldn't say that entirely because the whole idea of nitric oxide and some of these other things can have toxic side effects, but you don't have to have catalase, for example, because you don't have the peroxides being formed. You don't need superoxide dismutase because you don't have the superoxide anion being produced. So it's kind of, if you're going to live the anaerobic life, you have to have those traits that allow you to do it. If you're going to live the aerobic lifestyle, you have to have those traits to do that as well, which includes those detox enzymes. And so with, kind of looking at those things, I want you to realize that all of that is hardwired into the DNA of bacterial species. The things that you need downstream from respiration are as important as the redox chains that are doing it themselves. So you already know, because I've told you this enough times, that on Earth, the bacteria and archaea are the only things that are able to live an anaerobic life. Everything else requires oxygen as a final electron acceptor for respiration. But when we look at bacteria, realize bacteria are so flexible when it comes to their needs and requirements. And even an individual species of bacteria may have multiple ways to live their life. They could be both aerobic and they could be anaerobic. And so this is a characteristic that is very useful when it comes to classifying bacteria. And we did several tests. We did the catalase, oxidase, TSI, nitrate reduction test in the laboratory to try to figure out what these guys were doing, what they were doing with regard to their requirement or lack of requirement for oxygen, whether what I called living the aerobic or the anaerobic lifestyles. So you remember, hopefully also from lab, that the way we classify these as groups, the aerobes versus the anaerobes, right? So aerobic types of bacteria. They could be obligate or strict aerobes, which we are. Okay, so if we wanted to put us humans into that category, we would be considered strict aerobes because our primary means of producing ATP is through the electron transport system with oxygen as a final electron acceptor, with a few other things thrown in, but nonetheless, that would be us. So in a bacterial sense, if you grow bacteria in a broth called thioglycolate, which is this really dense, gross smelling stuff, but at the top of it, you could also do this in a soft agar. At the top of it, you'll have high oxygen, or 20, the normal percentage of oxygen. At the bottom, there will be low to no oxygen. So aerobic environment, anaerobic environment. And we did this when we used our stab medium as well. If you remember, we stabbed them to the bottom. If they can grow right to the bottom, then they must have what I called anaerobic tendencies. So the aerobes are the obligate aerobes. Notice they're growing there fully using oxygen as a final electron acceptor. So aerobic respiration is their metabolic pathway. And then microaerophiles also use aerobic respiration. But interestingly, they're not well equipped to deal with the reactive oxygen species, so they can't take it at full strength. And notice that they grow at lower or reduced oxygen concentrations. So that requires specialized incubation strategies, right? So if you're going to grow a culture of these microaerophiles, you actually have to provide for them a reduced oxygen environment. So that makes them very difficult to grow in a laboratory that isn't equipped for that kind of thing. So those are our two groups that make ATP by aerobic respiration. And then there are this spattering of anaerobic bacteria. So these are the ones that can live anaerobically. Obligate anaerobes live only anaerobically. They survive by using anaerobic respiration for metabolism. So to make ATP, they have an electron transport system where they hand those electrons off to something that isn't oxygen, one of those other gases. And then we have also these anaerobes. So in other words, they are living their life anaerobically. They are anaerobes at heart, but they are able to grow aerobically, because although they're primarily anaerobic, they also may have other pathways like aerobic respiration or fermentation, which allows them to grow in many different types of environments. So those would be the facultative anaerobes, which actually grow very well when there's oxygen because they have those detox enzymes. So they're cool, growing aerobically or anaerobically. And then there are the aerotolerant anaerobes, which are just like, I don't like it, but I'll put up with it, meaning they don't have ability to deal with reactive oxygen species very well. So if you remember, we did the test, a catalase test, which differentiated between these two groups. So these guys have catalase, so they can deal with those reactive oxygen species. So they live wherever they want very happily. These guys don't have catalase and therefore are stressed out when they are in an oxygen environment. They grow, but they grow slowly and they don't grow. That's not their best state, but they are at heart anaerobes. So when you look at the different environments for these things, in an environment where there is oxygen, in the bacterial world, oxygen present, when it is present, obligate aerobes are able to grow, but so are facultative and aerotolerant anaerobes, and to a certain extent anyway, the microaerophiles. If you have an environment where there is no oxygen, you can grow obligate anaerobes, facultative, as long as they're anaerobic. But remember, these guys, who are these guys? They are bacteria or archaea. The obligate aerobes, all other groups of living things, right? So the plants, the animals, the fungi, the protozoa, the protists, all fall into this category, whereas bacteria and archaea could be you know, they could live in any of these different environments, which means our own particular biases with regard to where we thought living things could live, where we thought stuff could live. Like we never thought, no, nothing can live under the ocean. No, nothing can live underground. No, nothing can live in outer space. No, nothing can live on other planets. Bacteria can. So it's something to kind of keep in mind as we think about astrobiology, which we're not going to, so don't anybody worry about that, okay? So these are the groups that we did those tests to find. I will have to say that in our laboratory, we are able to grow obligate aerobes, and we're able to grow either facultative or aerob tolerant anaerobes because of our culture conditions, the way we actually do it, the way our incubators are set up. And so when you did your human skin microbiome project, realize that your skin bacteria, because of the way we designed the experiment, the way we did our tests, were either aerobic, they're obligate aerobes, or they could have been one of these two, because we grew them in an incubator with full out oxygen. So, you know, it was one of those three, which by now you actually know what yours was. Okay, so oxygen is one of those chemical things that is you know, we have to make sure we match what the bacteria want in order to grow them successfully. Another one is temperature. So notice that life exists over several temperature ranges. So I'm not going to say most because I don't actually believe that, but the mesophile, and remember PHILE means to love. So the moderate temperature lovers are like us, and you'll notice that the rate of growth, you know, mesophiles can grow at colder temperatures, but they have a peak rate of growth at about 37 degrees Celsius, which is our body temperature. And there are lots of things, animals and you know, lots of different, everybody that grows in association with things that like that temperature would fall into this category. When we grow bacteria in the laboratory, we are growing mesophiles because why? We incubate them at either, you know, between 25 and 35 degrees Celsius. So that is, we are enriching, if you will, for mesophilic types of bacteria. But realize that there are psychrophiles and thermophiles, and then there are the hyper or extreme thermophiles, which, as you might expect, the thermophiles love to grow where there's heat. Notice the peak temperature is 60-ish, 60 degrees Celsius. So these grow best at temperatures above 55 degrees. And they include bacteria, but a lot of archaea are able to grow at those higher temperatures. And then there are the extreme thermophiles, which are archaea only, as far as we know at this moment, that have been found to survive at temperatures as high as 120 degrees Celsius. So just to tell you, when we autoclave our waste, microbial waste, before we are able to dispose of it, we autoclave it at 121 degrees Celsius for the purpose of sterilizing the waste. So these are bacteria, these hyperthermophiles are bacteria that would grow happily in the autoclave at those temperatures. So, and again, they are archaea, they're a subgrouping of archaea, and not any bacteria, or at least as far as we know at the moment, okay? So the psychrophiles like it cold. There are algae that are cold lovers, there are fungi that are cold lovers, but notice, and there are bacteria, by the way, that are psychrophiles that like it cold. Their maximum temp about 15 degrees Celsius, which is pretty darn cold. Remember, 0 degrees Celsius is 32 degrees Fahrenheit, just so hopefully you remember that little transversion. And then mesophiles, right, 98.6 or 37 degrees, 55 degrees, and then the hyperthermophiles that grow at temperatures, you know, that nothing else would be able to survive. Why? Because heat denatures stuff, right? As you know, heat causes proteins to denature, it activates enzymes, disrupts structures, So these are some pretty remarkable microbes that have some really fantastic adaptations that allow them to survive at those higher temperatures. So I just wanted to point out a couple of places where we find thermophiles and extreme thermophiles. Thermus aquaticus is the first thermophile to be discovered. It was discovered by a microbiologist named Thomas Brock, and it was back in the 1960s. And in the 1960s and 70s, by the way, there was the discovery that, holy crap, microbes can live at these temperatures. So Brock was considered, you know, one of these pioneers who said, you know what, there's microbes that are, their microbial metabolism is occurring in these places. This is the Grand Prismatic Spring at Yellowstone National Park, where the water temperatures are so high, they tell you, you know, if you've ever visited there, there's all these signs saying, don't go near the water because it's scalding or boiling in some cases. And the water looks perfectly clear. And you would think nothing can live there. It's rock with water in it. But guess what? All these pretty colors that you're seeing around the edges are microbial due to microbial growth. Many of them are photosynthetic, right? So this is one place. Why I pointed out Thermus aquaticus, by the way, is because after he discovered this, this culture of bacteria was used for a procedure called polymerase chain reaction, or PCR, and which revolutionized the field of molecular biology and genetics, totally revolutionized the entire field, because now we could amplify DNA from sources, and the rest, as you know, right? So if you can go grab DNA, you don't have to even look at where the DNA came from. You can just go grab it, amplify it, and then sequence it or do whatever you want with it. So for that process, it required an enzyme that was heat stable that would copy DNA. And it turns out the DNA polymerase from Thermus aquaticus was that enzyme. And so this really whole long, very interesting history, if you feel like doing a little bit of side research, go look up Roche, which was the company that developed PCR. At first, they patented this organism. The first time a living thing was patented by a company because they wanted the enzyme from that company, meaning nobody else could grow this particular bacteria because it produced this enzyme that was part of their process, this polymerase chain reaction process. So the creator of that process was Kary Mullis, but you know, there were lawsuits and lawsuits and lawsuits trying to prevent a company, a commercial entity, from patenting a living thing, which Roche won that. So interesting side thing that you can go look at. So thermophiles, extreme thermophiles, this is a deep sea thermal vent. It looks like a chimney, and all this black smoke you see coming out. So imagine this is 2 miles to 3 miles underneath the ocean at these areas of high volcanic activity, right? So the hot gases from underneath the mantle of the earth are spewing into the water. And that's what these clouds are. This is clouds of hot gases, right? Mineral rich gases, inorganic chemicals, meaning bacteria that are lithotrophic can use the electrons that are associated with these hot minerals and turn that into ATP, which then they can use, of course, for autotrophic means. So this also came from the 70s after Woods Hole Oceanographic Institute was able to develop a submersible submarine that was capable of going down that far. The intent was to just see what was at the bottom of the ocean. And honestly, the bottom of the ocean was relatively unstudied because it was like nothing can live down there. There's no light, there's no energy, there's no, you know, nothing there. There can't be anything living down there. And when they actually went down there, they saw these chimneys with the hot gases coming out of them covered with these things, which are called tube worms, by the way. And by the way, if you're a fan of SpongeBob SquarePants, he had a friend in one episode who was a tube worm. And so these tube worms live these are heterotrophic organisms that survive at these deep sea locations. This was a newly discovered living thing. And they survive because guess what? They're filled with lithotrophic bacteria that can turn this into ATP and then fix CO2 to produce biomolecules. So these are the autotrophs of the deep sea world, not light dependent, they're chemical dependent. So, and that's how they're doing it. So that's the basis for a whole food chain at the bottom of the ocean. Much more to come on that, hopefully. Okay, so then looking at salt concentrations. So realize that most cells have a tolerance of about 1% dissolved salt in water before osmotic pressure gets to be too great, right? So that's referred to as isotonic, which is about 1%, or it's 0.9% roughly. But when cells with this, and that's due to the cell membrane, right? Pressure on one side versus the other of water. So if the concentration is not even of water on either side, water will either flow into or out of the cell. So the non-halophiles, halo means salt, phile means to love, are the ones who are resigned to live in environments where the salt concentration is roughly 1%. So, and then, you know, as the salt concentration increases, of course, this is what happens. The cells shrivel up, the cells die. And then looking at there are moderate halophiles. So marine environments have a higher concentration of salt, and some bodies of water have very high concentrations of salt, like the Dead Sea, for example, which was called the Dead Sea because the first people that saw it were like, oh, nothing's living here because it's too salty, everything's dead, so it's the Dead Sea. So these marine organisms from 3 to 10% salt, right? So from 1% to 3 to 10% salt in water. And then there are the extreme halophiles, which can live up to from 30% on up to almost 100% salt. So how they are doing that is, again, through genetic adaptations that allow them to withstand all of those nasty forces of nature. And this is an environment where you could expect to see them. So these are tidal pools they're called salt flats, which is how salt is, sea salt, if you ever wanted to know, comes from. So they let the ocean flow in over these pools, close them off, and then allow the water to evaporate, which increases the concentration of salt over time. And so notice all the different colors, right? The purples and the greens and the oranges and the reds are due to the growth of halophilic bacteria, which are photosynthesizing. So they are able to produce their own food using that process, living happily there without any competition because nothing else can survive at concentrations of salt that are that high, except for these archaea, which are these extreme types of halophiles. So again, these are remarkable in their ability to survive in environments that are not, that, you know, up until the 1990s, people would say there cannot possibly be anything living in those locations. So not only, by the way, are these archaea heat and salt resistant, they are radiation resistant because you've got sun beating down on them on the tropics that are allowing for evaporation of the water, but also causing, you know, think about that, laying there baking in the sun for as long as the sun is up. So pretty remarkable microbes. And then the last consideration is pH. So as I said, bacteria and archaea can grow across the entire pH range from the, you know, very acidic to very alkaline types of conditions. So, you know, neutrophiles include a lot of other groups of living things, including humans and plants and fungi and, you know, algae and protozoa. But the acidophiles, the acid lovers, grow best at very low pHs, right? pHs of 2 to 3. And oftentimes they are in those environments, and as a result of their metabolism, they actually produce these acids. So not only are they able to survive in the environment, but they're also producing it. Like, for example, bacteria that reduce sulfate can produce sulfuric acid as a product of that. And that decreases the pH of the environment they're in. So if you're going to do that, you need to be able to live in those very acidic environments. And, you know, some types of fungi are able to grow at lower pHs, right? So they tend to be a little more acidophilic also. But then we get into the neutrophile range, which is the majority of the bacteria that we work with in labs that are symbionts of many of different types of animals. And the alkalinophiles are those that live where the pH starts to get higher and higher, right? So these are places where there's a influx of ions that lead to a raise in the pH, such as soda lakes, which are called soda because of baking soda, because of the bicarbonate that's in them. But that causes a higher pH, and there are bacteria in archaea that are able to grow in those environments too. So just a couple of examples here again. These are bacteria, as I mentioned, acid drainage. What that means is acid coming out of the ground when coal mining is being done. And this was an environmental concern and a coal mining concern for a long time, which it still is, but they've been able to mitigate it. So the acid drainage is a result of bacteria and archaea that are doing, like I said, for example, that using sulfate as a final electron acceptor and producing sulfuric acids or other types of acids. So that's what you see, all this reflective stuff, those are bacteria growing in those environments. And then over here on the alkalinophile side this is a farm where what they grow is spirulina. So spirulina is sometimes called blue-green algae. It is not an algae, it is a bacterium. It's a cyanobacteria, just so you know. And they are photosynthetic. So you have to grow them in a lake that is one of these soda lakes. You have to grow them in an alkaline environment because that's the environment where they grow best. So this is a farm for this bacteria, spirulina, which you can see is very, very green because these bacteria have chlorophyll, and they are farmed because spirulina or chlorophyll is a nutritional supplement that you can go to your health food store and buy, which apparently gives you superpowers if anybody's interested. So obviously all of these different concerns, all these different environmental conditions have to be met. Bacteria and archaea they live in the environments where those conditions are met. We live in an environment where our physical and chemical conditions are met for us. And so it's just that when we talk about bacteria and archaea, the range of possible environments where we can find them, I want to say, is limitless, because we have found them in practically every place we have ever looked, realizing that now we're not necessarily looking for the cells themselves, the bacteria themselves. We're looking for their DNA, and we found DNA and new species pretty much everywhere we've actually looked. So also, just kind of go along with this, I've provided you with a couple of other YouTubes for you to watch. So please go watch Dr. Garth Ehrlich and Dr. Bonnie Brassler talk about biofilms, very important aspect of bacterial life, especially as it pertains to medicine. And then go hear a little bit about the dark web of the carbon cycle, which is the video that I showed you on your schedule, just so that you can appreciate the remarkable diversity in terms of lifestyles that bacteria can have. So thanks, and I'll see you next time.